Today we're going to be talking about Chapter 8, Section 2, which is all about the structure of DNA. So before we get started, make sure that you have the correct Power Notes sheet. There is a concept map for you to arrange your information, and then of course several diagrams for you to label. So if you don't have this handy, go ahead and make sure that you download that, and then you can continue with the rest of this video. So we're going to start out by just a reminder of the structure of DNA. You already know that DNA is made up of a long chain of nucleotides. The nucleotide is the building block of a nucleic acid. And each nucleotide has three parts. Hopefully these are familiar to you. There is a phosphate group, which is generally represented by a circle. There is a sugar, which is going to be a pentose sugar or a five-sided sugar. And then there's always some kind of nitrogen-containing base or nitrogenous base. If we were to look at the molecular structure of this base, there would be nitrogen atoms in it, hence the name nitrogen-containing base. Now we do need to look at this sugar. The name of the sugar is deoxyribose. That's important. DNA stands for deoxyribonucleic acid. So the name DNA comes from the name of this sugar that makes up its nucleotides. Remember, RNA, ribonucleic acid, is going to be the other kind of nucleic acid. We'll learn more about that a little bit later in this unit, and it has a different sugar. So knowing the name of the sugar and how it's different between DNA and RNA is definitely an important concept for this unit. Now, the difference between the nucleotides that make up DNA comes from those nitrogenous bases. There are four different kinds of nitrogenous bases in DNA. They are thymine, cytosine, adenine, and guanine, abbreviated T, C, A, and G. Now notice how we have these organized into two categories. We have on the left the pyrimidines, and these are single ring structures. Well, what does that mean? That means if you look at the molecule, there is one hexagon or one closed ring of molecules here, or atoms in this molecule. Same for cytosine. Whereas a purine is a double ring structure. There's a hexagon and a pentagon, so that's two rings that are bonded together. Now you may be thinking, wait a minute, isn't there another base in RNA? And you would be right. There is. Uracil is the fifth base. Now, it's not found in DNA, so it's not a major focus of what we're talking about today, but it is an important base that you would find in molecules of RNA, and it is unique to RNA. It's only in RNA, not in DNA. It has a single ring structure, so it would be considered a pyrimidine as well as cytosine and thymine. Now, the structure of DNA was a really important scientific problem, and solving that problem, what this structure looked like, that represents years and years of work done by a lot of really prominent scientists at the time. And the two scientists that got there first, the ones that really figured out this structure and built the very first accurate model of DNA, are James Watson and Francis Crick. They're pictured here in this picture with their actual model that they built. And when they built their model, the key thing was realizing that DNA was a double helix. Double helix meaning two strands that are wound around each other, kind of like a ladder if you were to twist it into a spiral. And that double helix was made up of a backbone or two backbones. The two sides of the helix would be made out of sugars and phosphates, and then those nitrogen bases are going to connect on the inside of the DNA molecule. And we'll look at a diagram of that in just a minute. But Watson and Crick were not able to figure this out all on their own. They did build their model based on the work of some other scientists, so let's mention those as well. The first of these scientists that we'll talk about is Rosalind Franklin. She was working in a nearby lab, and she was an X-ray crystallographer. There's actually a picture of her. Let's go ahead and look at that picture. And to her right is an image that she took. This was produced by shining X-rays at a purified molecule of DNA. And if you're an expert in X-ray crystallography, when you see this shape, you know that that is the characteristic shape of a double helix. So she took this photo, and a colleague of hers who worked in the same lab took the photo. Rosalind Franklin actually didn't give permission or even know that this photo had been shared, but her colleague shared the photo with Watson and Crick. As soon as Watson and Crick saw it, they recognized what this structure meant and what it meant for their model of DNA. And that was a really key aspect for them to solve this problem of determining the structure of DNA. Rosalind Franklin, because she worked with X-rays, unfortunately she had cancer and she died at the age of 37. Watson and Crick did go on to win the Nobel Prize for discovering the structure of DNA, and even though Franklin's photo was critical in discovering that structure, she had already passed away at the time the prize was awarded, and by rule you cannot be awarded a Nobel Prize after your death. So she was not named in the prize, although Watson and Crick and Rosalind Franklin's colleague, the one that shared the photo, Maurice Wilkins, all three of them shared that Nobel Prize. Now, they also used the work of Erwin Chargaff, and you see him mentioned here as well. And he was a little bit earlier studying the structure of DNA, and he was able to measure the quantity of the different bases. And what he noticed was that the amount of adenine always equaled the amount of thymine, and the amount of cytosine always equaled the amount of guanine. And when Watson and Crick took that into consideration, once they knew that DNA was a double helix, it became obvious to them that if there were two strands, if there was an adenine on one side, there must be a thymine on the other side, and that would be why they were always found in equal amounts. So that has become what we call our base pair rules. And the base pair rules, there's two of them, because there's two different ways that these bases pair up. The first one is that adenine always pairs with thymine. So if you have an adenine, which you see here in green, on the opposite strand would be a thymine, and they pair up with each other. We would call that complementary. We would say adenine is complementary to thymine. And then the second base pair rule, cytosine is always going to pair with guanine. So those two would be complementary. You can see guanine is shown in purple and the cytosine is shown in yellow, and they're always opposite each other. Now, if you are thinking about the structure here, it's interesting to note that we always have a single ring paired up with a double ring, and that's just to make sure that the double helix stays a uniform width. If you had two double rings paired up with each other, it'd get really wide in that one area. Two single rings, it would be too narrow. So it always has to be a single ring with a double ring. Now let's talk a little bit about this structure of DNA. So we're using a different diagram here. We're looking at the nucleotides and how they pair up. So here would be one nucleotide. We have a sugar, we have a phosphate, we have a nitrogen base. It's pairing up with the nucleotide on the other side, sugar, phosphate, nitrogen base. And notice that we have two different kinds of bonds. The backbone, which would be these two sides, right? There's the backbone, there's the other backbone. So it's not one backbone, it's two backbones because it's a double helix. These are all held together by covalent bonds. They're solid lines. The bases, which holds one strand to the other strand, these are dashed lines, which you probably remember represent hydrogen bonds. So these individually are very weak bonds, but look, we have two here, three here, two here, three here. If we have thousands of nucleotides all hydrogen bonded together, that becomes a strong enough force to keep DNA intact with both strands attached to the other. And even though we have hydrogen bonds in the center, that is enough to keep the molecule together. So now let's talk about the direction of a DNA strand. This is really important for the way that the molecules work when DNA is being read and copied or being used to make a protein. The direction is really important. So scientists have actually numbered the carbons so that if they talk about carbon number 1 or carbon number 2, another scientist knows exactly which carbon they mean. Because the numbers are always going to start with the first carbon going clockwise from the oxygen. Well, what do I mean by that? Here's our nucleotide. There's oxygen up here. If you think of this as a house, the oxygen's always on the top of the roof here. So here's our carbon number 1, here's our carbon number 2, carbon number 3, carbon number 4, and carbon 5 is up here. So if we have numbered these carbons, it's really easy to identify which carbon we are talking about. And the carbons that are going to be important are going to be the carbons that are involved in this backbone. So notice this 5' carbon has the phosphate group attached. Where does it attach to the next nucleotide? It attaches to its 3' carbon, right? So if I were to take another molecule just like this and I wanted to pair it up, the phosphate group would attach to this carbon down here. That's our 3' carbon. And if we look at the entire strand, these are 3 nucleotides all paired up with each other. The top of the picture is where the 5' carbon is, right? All the 5' carbons are on the top. So we would say this region, if we looked at this strand as one piece, the top would be the 5' end, and the bottom, because that's where all the 3' carbons are, that would be our 3' end. So let's look and see what that means if we go back to that original picture of DNA here. So we know that these DNA strands are paired up with each other, but they're actually anti-parallel. So anti means against, right? So if they're anti-parallel, that means one's going in one direction and one is going in the opposite direction. So this is our 5' end on this strand. How do I know that? It's the side where the phosphate is. The phosphate is always attached to the 5' carbon coming out of this sugar here. So this is our 5' end. So there's one nucleotide. This is a 3' end. There's the 5' end of the next one. So all the way at the bottom would be the 3' end. All of the 3' carbons are on the bottom here. All of the 5' carbons are on the top. So this strand is 5' on this side, 3' on that side. Well, look over here. Now, all of these nucleotides are pointing the opposite direction. All of our houses are upside down now, right? So that means that the top is now our 3' carbon, and the bottom is our 5' carbon. Our 5' carbon, again, is always the one where the phosphate group is attached. So this strand goes from 5' to 3'. This strand goes from 3' to 5'. So again, we're really just looking at these numbers as a way to tell which direction we are reading the molecule in, because the way that the enzymes work for DNA synthesis and transcription and translation is that they are only going to be able to work in a single direction. So this is how we identify the direction in a DNA molecule. So this is the end of Chapter 8, Section 2. I just want to leave you with the section quiz here from your textbook. Take a look at these 5 questions, see if you can answer them, and we will discuss the answers to this in class. So hopefully you found this to be a helpful presentation, and if you have any questions, please make sure that you ask those.